close your mouth. Clench your back teeth. Hard. Feel the slab of muscle that bunches up just above your jawline in front of your ear. That is the masseter. Pound for pound, the strongest muscle in your body. Every time it contracts against resistance, it fires a signal up the fifth cranial nerve, across the brainstem, into a curved structure deep in your temporal lobe called the hippocampus, the structure that stores memory. For 3 generations, that signal has been getting weaker. And a quiet line of research out of Japan, Harvard, and a handful of neuroscience labs in Italy and Sweden has spent the last 25 years working out what that silence is doing to the brain. Run your finger from the corner of your mouth to the front of your ear. Clench again. You just trace the attachment line of the masseter. Higher up, under your temple, sits its partner, the temporalis, a fan of muscle that wraps the side of your skull. These 2 muscles close your jaw. Together, in a healthy adult, they can generate a sustained bite force of over 160 pounds on the molars. Every tooth in your mouth sits inside a thin, flexible sleeve called the periodontal ligament. The ligament is about 0.2 of a millimeter thick. Inside that sleeve are thousands of mechanoreceptors, nerve endings that fire the moment the tooth is loaded. Each receptor is wired into the trigeminal ganglion, a dense bundle of nerve cell bodies buried behind the cheekbone. From the ganglion, the trigeminal nerve enters the brainstem at the pons. The trigeminal is the largest of the 12 cranial nerves. It carries more raw sensory traffic into the brain than any other nerve in the head. More than your eyes, more than your ears, more than taste and smell combined. And most of its fibers come from one place: your mouth. A few millimeters from where the trigeminal enters the brainstem sits a tiny bluish cluster of neurons called the locus coeruleus. The name means the blue spot. It is about the size of a grain of rice. It is also the source of nearly every drop of noradrenaline in your brain. Deeper in each temporal lobe, is the hippocampus, shaped like a curled seahorse. The region that forms new memories, builds spatial maps, and is the first structure to atrophy in Alzheimer's disease. Here is the circuit. The trigeminal nerve talks directly to the locus coeruleus. The locus coeruleus projects upward into the hippocampus, which means every time you bite down hard on something, a signal travels from a tooth in your mouth through your brainstem into the memory center of your brain. Researchers call it the trigeminal locus coeruleus hippocampus axis, and it is activated almost exclusively by chewing hard food. The first mechanism, the signal that leaves your tooth when you bite down on a raw carrot, the pressure does not just sit on the tooth. It compresses the periodontal ligament underneath. Inside that sleeve, thousands of mechanoreceptors deform. When they deform, they fire. Each tooth is capable of producing a precise burst of electrical signal that encodes 3 things: how hard the bite is, in which direction the force is acting, and how long the load is sustained. This signal is continuous, not a single spike. A dense train of electrical pulses that update hundreds of times per second. If you are chewing an almond, your brain is receiving a stream of mechanical data as detailed as what your fingertips send when you read Braille. All of that traffic moves through the trigeminal nerve. Karen Kubo and Minoru Onozuka, working originally at Kanagawa Dental College and later at Gifu University and Nippon Sports Science University, spent much of their careers mapping this traffic. Their conclusion, across dozens of experiments, was that the trigeminal nerve during hard chewing is not a specialized sensory channel. It is a floodgate. It delivers more activating input to the brainstem per minute than any deliberate cognitive exercise a human being can perform. Now think about what happens when you stop loading the ligament. Soft bread, oatmeal, a smoothie, pasta cooked past al dente, a banana, yogurt, scrambled eggs, steamed fish. These foods still require chewing, but the pressure on the periodontal ligament is minimal. The mechanoreceptors barely deform. The signal that leaves each tooth is a trickle, not a flood. Over one meal, it does not matter. Over 10,000 meals, which is roughly 1 decade of eating 3 times a day, the trickle becomes the baseline. The flood becomes a rare event, and the brain wires itself around what it regularly receives. The person who has not eaten anything harder than a soft sandwich in 20 years is not just losing a sensory input. They are losing the single largest mechanical signal the brain evolved to expect, which is where the next mechanism begins. The second mechanism, the J blue spot wakes up. The signal from the trigeminal does not just pass through the locus coeruleus. It wakes it up. The locus coeruleus is, in a way that neuroscientists still find slightly strange, the main source of arousal for the entire brain. When the blue spot fires, noradrenaline is released simultaneously into the cortex, the hippocampus, the amygdala, the cerebellum, and the prefrontal cortex. It sharpens attention. It increases vigilance. It dilates the pupil. Pupil dilation is not an emotional response. It is a direct readout of locus coeruleus activity. This is one of the cleanest measurements neuroscience has. In 2017, a research group led by Filippo Tramonti Fantozzi at the University of Pisa published a study in Frontiers in Neuroanatomy. The design was simple. Subjects were given a cognitive task, retrieving specific target numbers from a grid. Their pupil size was measured throughout. Some subjects chewed a soft bolus during the task. Others chewed a hard bolus. A control group did not chew. The hard chewing group showed significantly larger pupil dilation during the task. They also retrieved the target numbers faster. The soft chewing group showed smaller effects. The control group showed none. The effect persisted for up to 37 minutes after chewing stopped. Tramonti Fantozzi's interpretation was direct. Chewing a hard bolus activates trigeminal afferents. Trigeminal afferents wake up the locus coeruleus. The locus coeruleus pumps noradrenaline into the cortex, and the entire brain becomes briefly more alert, faster, and more accurate. Soft food does not trigger this. The pressure threshold for periodontal mechanoreceptors is not crossed. The trigeminal signal never reaches the level needed to cascade through the blue spot. Here is the part that tends to land. The locus coeruleus degenerates with age. In Alzheimer's disease, it is the first structure in the brain to show signs of pathology, sometimes decades before memory symptoms appear. Some researchers, notably Mara Mathur at the University of Southern California, have argued that a healthy locus coeruleus in late life may be one of the best predictors of cognitive resilience. The trigeminal locus coeruleus axis is one of the most direct ways to exercise the blue spot from the outside. You close it every time you eat something soft, which matters for the next structure in the chain. The third mechanism, the seahorse and ites dendrites noradrenaline from the locus coeruleus, does not merely excite the hippocampus. It regulates the growth of new neurons inside it. The hippocampus is one of only 2 regions of the adult human brain where new neurons are reliably born throughout life. The process is called adult hippocampal neurogenesis. The cells are born in a thin curved zone called the dentate gyrus. They migrate a short distance, grow dendrites, and wire themselves into the existing memory circuitry. A human brain produces somewhere on the order of hundreds of new hippocampal neurons per day, possibly more in some people, possibly fewer. In animal studies, the rate of this neurogenesis is strongly influenced by 3 things: aerobic exercise, caloric restriction, and chewing. In 1999, Minoru Onozuka and his colleagues ran what would become a foundational experiment. They took a strain of accelerated aging mice, the SAMP8 strain, and split them into 2 groups. One group ate their normal hard pellet diet. The other group had their molars filed down so they could not chew effectively. Both groups received identical nutrition. The difference was purely mechanical. One group chewed, the other could not. Within a few weeks, the molarless mice showed significant loss of neurons in the hippocampal CA1 region. They performed worse on spatial memory tasks. Their dentate gyrus showed decreased neurogenesis. By late life, their hippocampal atrophy resembled the pattern seen in early Alzheimer's disease. In 2005, Kubo and colleagues published a paper in Brain Research showing that the Molarless condition also reduced the density of dendritic spines in the hippocampus, the tiny protrusions on neurons where synaptic connections form. Fewer spines, fewer connections, less plasticity. In 2010, Aoki and colleagues, publishing in Neuroscience Letters, confirmed that the Molarless condition in rats suppressed the proliferation of new hippocampal neurons in the dentate gyrus. The effect was not subtle. In human work, Hirano and colleagues at the National Institute of Radiological Sciences in Chiba, Japan, used functional magnetic resonance imaging to watch the human hippocampus during chewing. Their study, published in Brain and Cognition in 2013, found that chewing increased hippocampal blood oxygen level signal and improved cognitive processing speed. The activation was not in one isolated region. It spread across the middle frontal gyrus, the precuneus, the thalamus, the right premotor cortex, and the hippocampus itself. Not a single muscle, a system. So here is what the evidence says, stated plainly. In animals, reduced chewing causes measurable loss of neurons in the memory center of the brain. In humans, chewing measurably increases blood flow and activity in the same regions. The mechanism is consistent. The direction of causation is consistent. And the structure most affected is the hippocampus, which is the first structure that dies in dementia. Now layer one more finding on top. The fourth mechanism, the river off blood your brain, is 2% of your body weight. It consumes 15% of your cardiac output and 25% of your resting energy supply. It is the most metabolically expensive organ you have. And it has essentially no fuel storage. Every minute it needs a fresh delivery. Cerebral blood flow is not uniform. When a region is working harder, local vessels dilate and flow to that region increases. This is the principle that makes functional imaging possible. During hard chewing, regional cerebral blood flow increases by roughly 25 to 28% across the prefrontal cortex and sensorimotor regions. The number comes from positron emission tomography studies and functional magnetic resonance imaging studies performed over the last 2 decades, including the Hirano group's work and independent replications. Think about what this means mechanically. A 1/4 increase in blood flow to the brain is not a minor shift. It is what a moderate jog produces. And the jog has to last 10 or 15 minutes before cerebral flow adjusts to it. Chewing produces the same response in 30 seconds. No supplement does this. No nootropic pill produces a rapid 25% increase in prefrontal cortical blood flow on command. The body has a mechanism built into its anatomy for delivering a surge of oxygenated blood to the brain on demand, and that mechanism is triggered by loading the periodontal ligament against resistance. Soft food does not trigger it. The mechanical threshold is not reached. The person who eats mostly soft food is not only losing the trigeminal signal and the locus coeruleus activation. They are losing the brief daily surge of cerebral perfusion that the chewing reflex was built to produce. And this is before we get to the bone. The fifth mechanism, doit your grandmother's jawbone you said to look like bone, is not inert. It remodels constantly in response to the forces applied to it. This is called Wolff's law, named for the 19th century German surgeon Julius Wolff, who first formalized the principle. Load a bone. It grows denser and longer at the points of stress. Unload it. It resorbs. Your mandible, your jawbone, is one of the most mechanically responsive bones in the human skeleton. It grows in response to bite force. It atrophies in response to disuse. Daniel Lieberman at Harvard has spent much of his career on this question. In 2004, Lieberman and colleagues published a study in the Journal of Human Evolution in which they raised rock hyraxes on cooked versus raw food. The raw food animals generated masticatory strains in the jaw and face that were up to twice as high as the cooked food animals, and their facial bones grew accordingly. The cooked food hyraxes had approximately 10% less growth in the lower and posterior regions of the face, where the chewing strains are highest. 10%. on a short-lived mammal over a single lifetime. In humans, the pattern has been documented across archaeological time. A comparison by Juhani Varrela at the University of Turku, published in 1992, examined late medieval Finnish skulls and compared them to modern Finns. Same population, same genetics, different diet. The modern Finns had, on average, mandibles that were 6% shorter than their medieval ancestors. 6% in less than 1,000 years. This is a bone responding to what you do with it, not a genetic drift, not evolution, a developmental response. There is a more extreme version of this finding. In patients with significant loss of dental function, full dentures that fit poorly, long-term missing molars, atrophied masticatory muscles from disuse, the ramus of the mandible and the alveolar crest where the teeth sit can lose up to 50% of their height. A bone literally melting because no one is pulling on it anymore. Here is what is worth sitting with. The mandible is continuous with the temporomandibular joint, which sits directly below the temporal lobe of the brain. The muscles attached to the mandible insert along the base of the skull. The nerves that innervate the teeth run through the same foramina in the cranial base. When the jaw shrinks, the entire architecture of the lower face reorganizes: airway, sinuses, muscle insertion points. Obstructive sleep apnea, chronic nasal congestion, crooked teeth, impacted wisdom teeth, tongue thrust in children. These are not separate diseases. They are surface signs of a jaw that was never properly loaded during growth, and the load is missing because the food is soft. Weston Price, a Cleveland dentist who in the 1920s and 1930s traveled to remote populations eating traditional diets, noticed that the generation that switched to the Western diet showed dental crowding and narrow arches by the time they were adolescents. The previous generation, eating the same genetic inheritance but a harder diet, had perfect dental alignment. The change was not genetic, it was mechanical, and it took one generation. Bone listens to what the muscles tell it. When the muscles are quiet, the bone stops growing, and quiet bone means another system runs silent. The sixth mechanism, the cooling off, the stress response, the hypothalamic pituitary adrenal axis, is the circuit that releases cortisol during stress. It is coordinated from the paraventricular nucleus of the hypothalamus, which sits just above the brainstem. When it is overactive, cortisol rises. Chronic cortisol elevation is toxic to the hippocampus. Among other things, it suppresses the very neurogenesis we discussed 2 sections ago, which makes the next finding land harder. In a series of experiments led by Kinya Kubo, rats were subjected to restraint stress, a standard protocol for elevating cortisol. In one group, the rats were simply restrained. In the other group, the rats were restrained but given a wooden stick to chew on. The chewing group showed significantly lower plasma corticosterone levels. Their hippocampal neurogenesis was preserved. Their BDNF expression, brain-derived neurotrophic factor, a growth molecule essential for neuron survival, was higher. Their spatial memory did not decline. Same stress, different outcome. The only variable was the act of chewing under load. Andrew Scholey, working at Swinburne University in Australia, extended this into human studies. Subjects who chewed gum during a stressful cognitive task reported lower subjective stress, showed reduced cortisol, and performed better on attention measures. The effect was modest but replicable. The mechanism is now reasonably understood. The trigeminal locus coeruleus pathway releases noradrenaline at the same time it modulates corticotropin-releasing hormone in the hypothalamus. It acts as a brake on the stress response. When the brake is engaged by chewing, cortisol does not climb as high. A person who does not chew hard food is not only losing the memory-supporting signal, they are also losing a built-in regulator of their own stress physiology. Person over 50, who has been told for a decade that the way to manage stress is meditation or breathing exercises. Who has tried both, maybe found them helpful, maybe not. Who eats a soft breakfast, a soft lunch, a soft dinner. Who has not bitten into anything mechanically demanding since the 90s. That person has a stress regulator they have been unknowingly putting to sleep 3 times a day. A PAUs, what the evidency actually shows. Before going further, it is worth being honest about where the science is firm and where it is still assembling. The strongest claims are the animal findings. The Molarless mouse studies have been replicated dozens of times across multiple labs in Japan, Italy, and China. The link between reduced chewing and hippocampal atrophy in rodents is not controversial. It is textbook. The cerebral blood flow findings in humans are also well established. Functional imaging during chewing reliably shows a 20 to 25% increase in prefrontal and sensorimotor perfusion. This is measured, replicated, and physiologically unsurprising. The trigeminal locus coeruleus connection in humans is documented through pupillometry, imaging, and now several independent cognitive task studies. Call this strong evidence. The link between chewing, stress hormones, and cognitive performance in humans is supported by multiple studies, but effect sizes are modest and depend on the task. Call this moderate evidence. Real. Replicable. Not enormous. The long-term preventive claim that regular hard chewing across a lifetime reduces dementia risk is the least firm. It is physiologically consistent with everything above, but it has not been tested in a randomized controlled trial. It could not easily be. Epidemiological studies suggest that tooth loss and poor masticatory function correlate with cognitive decline. That correlation is robust. The direction of causation is plausible. The mechanism is laid out. But call this inference, not proof. And the claim about your own personal outcome, what will happen to your own brain if you chew differently starting tomorrow, is an extrapolation, a reasonable one, but an extrapolation. What the research establishes with high confidence is narrower and still remarkable. Chewing hard food generates a specific and measurable signal that activates specific and measurable circuits involving the brainstem, the arousal system, the memory center, and the stress regulator. Soft food does not generate that signal. A population eating progressively softer food across generations is silently dismantling a mechanism the brain evolved to depend on. Whether that dismantling, in your specific body, meaningfully changes your dementia trajectory 50 years from now, that nobody can tell you. Nobody should claim to tell you. What the literature can tell you is that the signal is real, the circuit is real, and the damage from disuse is real. The rest is your decision. The reframe, what the pill, is actually doing here, is where it becomes interesting. The global market for Alzheimer's disease medication in 2024 was estimated at approximately $8 billion per year. The most widely prescribed drug in that market is Donepezil, sold under the brand name Aricept. It works by inhibiting the enzyme acetylcholinesterase, which increases the amount of acetylcholine available in the synaptic clefts of the hippocampus and cortex. More acetylcholine means more signaling. More signaling means, in theory, slightly better memory function for some patients in the early and middle stages of the disease. The effect size is modest. Donepezil does not reverse the disease. It does not stop progression. In well-controlled trials, it produces a small improvement in cognitive scores that lasts 6 to 12 months before the underlying pathology overwhelms it. Side effects include nausea, diarrhea, vivid dreams, bradycardia, and in rare cases syncope. A daily prescription runs anywhere from tens to hundreds of dollars per month, depending on country and insurance. Now look at what chewing does. Chewing activates the trigeminal nerve, which projects into the nucleus basalis of Meinert, the cholinergic hub in the basal forebrain that sends acetylcholine into exactly the cortical regions donepezil is trying to enhance. It also activates the locus coeruleus, which delivers noradrenaline to the same memory circuits. It increases hippocampal blood flow by roughly 1/4. It preserves dendritic spine density in animal models. It lowers cortisol, which protects the very neurons donepezil is trying to preserve. In other words, The pharmaceutical industry has built an $8 billion business around a drug that approximates, through a single molecular pathway, one small piece of what chewing a raw carrot does to the brain through 5 simultaneous pathways. Be careful with that sentence. The body's version is milder. The drug exists because for a patient with moderate Alzheimer's, chewing a carrot is no longer enough. The disease has gone too far. The pharmaceutical dose is designed for a brain that has already lost most of the machinery this article is describing. But for a person in their 50s or 60s or early 70s whose machinery is intact, who still has the muscles, the teeth, the trigeminal traffic, the drug is not the first-line intervention. The drug is what you buy when the first-line intervention has been unavailable to you for 40 years because your food was too soft to trigger it. That is the reframe. Not that chewing cures Alzheimer's. Not that you can skip medication by eating almonds. Neither of those claims is true. The reframe is that a mechanism built into your skull, for free, powered entirely by the muscles of your face, has been quietly replaced by a molecule that costs billions of dollars and produces a fraction of the effect. Because the original mechanism required effort. And somewhere around the 1960s, the food system stopped requiring it. The H.E. Close, your grandmother probably said at some point, chew your food well. 30 times per bite. Slow down. Use your back teeth. She did not know about the trigeminal nerve. She did not know about the locus coeruleus. She had never heard the word neurogenesis. She had never read a paper by Onozuka or Lieberman or Kubo. She knew what she had been told by her own grandmother, which was what her grandmother had been told before that. Folk wisdom built across generations of people who lived in bodies that still worked. What she was saying, translated into the language of 21st century neuroscience, was this: load the periodontal ligament hard enough to fire the 5th cranial nerve, wake the blue spot in your brainstem, flood the hippocampus with noradrenaline, raise the cerebral blood flow by 1/4, preserve the dendritic spines, dampen the stress axis, keep the jawbone dense, and do it 3 times a day, every day of your life. She said it in 5 words: chew your food well. So, go find a raw carrot, an apple with the skin on, a piece of dense bread with a real crust, an almond, a piece of jerky, a radish, a stalk of celery, anything that fights back a little when your molars meet. Bite down. Feel the masseter clench. Feel the pressure load the tooth. Somewhere 2 inches deep, behind your cheekbone, the trigeminal is firing. A signal is traveling to the blue spot. The blue spot is releasing noradrenaline into your hippocampus. Blood is rushing into your prefrontal cortex. Cortisol is stepping back. Your jawbone is taking a load it has not taken in weeks. This is what your brain was built to do 3 times a day. It is still willing to do it. It is just waiting for you to give it something to bite into. Chew!